Okay, this is one of the spotlight presentations, so I will begin. And today, it's two speakers, which is me, Shambhavi, and Jian Peng, and we're going to talk to you about the work that GenScript does. Sorry, it's too much light straight on my face. Using the mRNA and LNPs for advancing gene editing solutions. So who are we? We are GenScript, and GenScript has been around for close to 20 years, and we are a life science service-based industry, and with a footprint across several different countries. And predominantly, since my talk is directed towards mRNA, the mRNA production and the R&D happens in Seattle area. And what we work on is not just providing linear mRNA, but we have linear, circular, and self-amplifying mRNA. There are three different product lines that we support with respect to mRNA. And not just process development, but also core R&D on the mRNA design and improving the quality and providing high-quality mRNA. In addition to mRNA, we also have LNP products, and I think there have been several talks about on mRNA and delivery of mRNA using LNP, so I'm not going to dive too much into it. But all our processes at GenScript are involved in using some of our proprietary ionizable lipids, as well as known lipids, which are SM102, ALC, LP01, and MC3. They are the predominant ones that we've used to streamline our processes. We also provide targeted LNPs that can help deliver your LNPs to targeted tissues or cells. And I think if you guys have attended several of the talks where people are developing mRNA-based drugs, one of the bottlenecks is streamlining the process for developing targeted LNPs. And because there are a lot of moving pieces in it, LNP is a complex process which involves multiple lipids. And post that, there's a conjugation process as well to tag the antibody, which is one of the process of developing targeted LNPs. And we do streamline that process considering each of the analytical steps to ensure that it's a robust process for developing conjugated, targeted LNPs that can target your specific tissue or organ. So what are we using it for? As I initially mentioned, we are trying to advance the gene editing solutions with coming up with optimized mRNAs that can enable gene editing. And one of the key processes, again, there are several ways by which you can deliver mRNA, and either with viral vectors or with non-viral methods, which is LNP. And one of the limitations of using viral vectors are that they are highly immunogenic, and there's a size limitation for the cargoes that can be put into it. So whereas LNP has no size limitation, and you can actually redose it, and it is immunogenic, but the toxicity can be reduced by process optimizations. So we are using LNP and mRNA for CRISPR-based systems and other systems as well that I'll talk about in performing gene editing. There are two components to gene editing, is that you do a knockout, or you knock down a gene, or particularly you can also knock in a gene or a target DNA into it. I'm going to talk about that, but prior to that, one of the key elements, again, is the mRNA. And what at GenScript we have done is that we have improved the quality of the mRNA by optimizing the UTRs. And also NLSs in the mRNA, because all these mRNAs that are, or proteins that are being utilized for gene editing have to be shuttled into the nucleus. So how many NLSs are important? How do you improve the expression of the mRNA? Because you have to appreciate the fact that when you're making the mRNA, when you're translating the protein, the guide RNAs are sticking around, and you have to have enough bolus of the protein and the guide RNA sticking around for it to shuttle into the nucleus. So we have to optimize the amount of the expression of the mRNA. And we did that. We screened several different UTRs, and we were able to identify one that has higher expression in liver, not just liver, but other organs as well. We initially wanted to target it to a particular tissue, but that's not how science works. You intend to move in one direction, and it takes you in the other. So we were able to identify a specific UTR that has higher expression than known 5' UTRs in the mRNA, which is quite exciting. And in addition, GenScript also has an AI-dependent tool that can help you design mRNA, codon optimize your mRNA to enhance the expression. And we have used that for most of the gene editing mRNAs. And as you can see, for prime editing, which is one of the proteins that is required for prime editing, which is PMAX, we used the codon optimization tool, and we were able to not just enhance the expression, but also increase the editing percentages in cells. Moving on, as I mentioned, one of the moving parts in gene editing is either knockout or knock-ins, and we've have had success with generating knock-ins in cells. And we've tested both short DNA, donor DNA encapsulations and longer DNA, donor DNA encapsulations using LNP. And there are several ways by which you can deliver LNP. Because it's a 3-component system or a 3-payload system, where you have the Cas9, you have the guide, and you have the donor DNA. So either you all encapsulate all in one LNP, or you co-deliver them. And we tested both these processes, and we discovered that co-delivery significantly enhanced the knock-in efficiency, achieving around greater than 60% efficiencies with LNP. And with the longer donor DNA, as you know, that cells are in the repair mechanism, it does HDR, which is homology-directed repair, and the efficiencies are on the lower end with the longer donor DNA. So we also tested several different components for the longer donor DNA, and what we have figured is that we tried the single-stranded DNA, which is a known donor DNA that people use for gene knock-ins, and we compared it with circular single-stranded DNA, and that significantly improved the knock-in efficiencies, as you can see. And we were able to get around 20% knock-in efficiencies with longer DNA as long as 2 kb, incorporating EGFP within the genome. So for furthering the processes, one of the key components for CRISPR studies is in vivo CAR-T. Either you can have an CAR mRNA, or you can actually generate knock-ins, right? So before you do that, you also have to have knockouts and efficient knockouts. So using our guide RNA and the mRNA modified ESP-Cas9 mRNA, we were able to achieve high editing percentages at the TRAC locus, achieving around greater than 80% efficiencies in primary human T cells and Jurkat cells. And in addition, we've also tested several different mouse cell lines for editing efficiencies. And as you can see with Cas9, you have to test out several different guide RNAs, because each guide RNA has a different efficiency. And we also have a platform for guide RNA design, and that can enable you to design highly efficient guide RNA that can have higher on-target percentages versus off-target. As I've mentioned that we work towards optimizing the LNP formulations, it can make a huge difference in the editing percentages or the potency of the mRNA that is delivered. So we optimize the LNP compositions to target one of the clinically relevant genes, which is PCSK9, and we tested its editing in liver cells. We started out with SM102, which is a known ionizable lipid, but we modified the compositions with either a helper lipid or another ionizable lipid. Each of the ratios are specifically changed or optimized to give you editing percentages, and that requires a lot of screening. So we screened a lot of several different formulations, and we were finally able to zone in into 2 of them, as you see, one with the helper lipid and the other with the another ionizable lipid that can help you improve the editing percentages. So not just working with the Cas9, we have also developed formulations that can enable you to perform base editing. I'm sure all of you know how base editing works, but as I talked about Cas9, which can generate a DNA break within the genome, this does not create a double-stranded break. It has a nickase and a deaminase domain that can make mutations, that can correct mutations. And GenScript has proprietary formulations that have shown very high higher editing efficiencies across different cell lines, it can be easily utilized for editing, for base editing as well. In addition to base editing, we have worked on prime editing applications as well. And prime editing is, again, a different modality or module of genome engineering. And rather than having, again, this also doesn't create a double-stranded break, but it has a lot of moving parts to it. It has the prime editing enzyme that is a combination of reverse transcriptase and a nickase, and it helps make edits. And the prime editing requires a PEG guide RNA, which is a larger long RNA, and a nicking guide RNA. And so at GenScript, we have a process that can make really long guide RNAs, around 200 nucleotide with high purity, and can be used for downstream applications. So we have also developed, GenScript has a modified SM102, which is its proprietary LNP that can enable very high prime editing in HECT93 T cells. Now, HECT93 T cells are easy cells to modulate or make changes to. We have made further changes in the mRNA quality and also the LNP compositions that can enable you to target different cell types. Prime editing is slightly different than Cas9. Cas9 is quite robust, and it can work across different cell lines easily. But with prime editing, that requires a lot more optimization. And as you can see, as I initially talked about, we changed the NLSs. We've also tested how many NLSs are important for prime editors or other editing enzymes as well. And the RNA's H domain, whether that's necessary or not. And those optimizations gave us higher editing efficiencies if you have 4 NLS versus 2 NLS in the main mRNA or the protein. We have a proprietary LNP that can enable editing in liver cells. And what was interesting is that we tested several different PEG guides. You have to screen with prime editing. One of the key components is PEG guide, and the design of it is crucial, and that requires a lot of screening. What we've learned from this is that the editing percentages are dependent on what kind of mutation you want to make or edit you want to make. For example, if you are trying to generate a deletion in the ORF, you might not be able to get very high editing, versus if you're doing a single base substitution, you will manage to get higher editing efficiency. So in liver cells, we were able to, in vitro, we were able to get 40% editing. We've also been able to achieve or target T cells, and at this point, we have lower editing, and we're working towards improving the editing percentages in T cells using prime editing mRNA. This brings to an end to my presentation, the first half of it. And again, I would like to reiterate or summarize that we provide one-stop solution from GenScript provides a one-stop solution, starting from plasmids to the mRNA, and we can provide several different mRNAs for your therapeutic purposes and that you can utilize in your research. And the other factor is if any of your guys' research aligns or if it's of interest to you, because we would like to collaborate with people who, where we can help them accelerate their research work. That brings to the end, and if you have any questions, I would be happy to take. None, I guess. I will give the mic to Jianpeng, who's going to talk about the guide RNA synthesis.
Thank you. So, hello everyone. This is Jianpeng Wang, and I'm the head of the R&D department for nucleic acid and the peptide. So I'm going to take the second half of the presentation is the securing CRISPR pipeline with scalable guide RNAs. Oops, it doesn't work. Yeah, sorry. Would be divided into 3 parts. First, about the guide RNA in diverse CRISPR systems, and then the evolution of the regulatory requirements, and we'll give some case studies. And the guide RNA in diverse Cas systems is that this is the Gen CRISPR solution that we target to offer services to knockouts and knock-ins. Generally, we provide the guide RNA services to all kinds of Cas systems, including Cas9, Cas12, Cas13, and also the base editing and the prime editing. Also, we have the Cas enzymes in both formats of the mRNA and also the protein formats. For knock-ins, we have templates such as the double-strand knock-in templates, single-strand, and also the circular double-strand. We call it GenCircle as knock-in templates. Specifically for the guide RNA, we can synthesize up to around 240 mer using pure chemical synthesis step. That this can, accelerate all the pioneering work especially for the prime editing. And to support the clinical trials, we designed our services in 3 hierarchical services, including the research use only, the R&D edit, and also the cGMP. For the research use only, we have the dissolved version and the HPLC purified version to support the early discovery phase and the preclinical, also the process development work. For R&D edit, we are targeting to support the preclinical services and also the R&D application. And for all the GMP services, we can cover all the clinical phase 1 to all the way to the commercialization. And these 3 categories, we have clear definition in terms of the application scenarios, quality management systems, and also the analytical methods, kind of everything included. And what's more, that we can, we have already streamlined the GMP manufacturing to shorten to 1 month. This is including also the manufacturing, QC, and QA releasing. And this is our manufacturing site. We have 400,000 square feet cGMP manufacturing space with 8 manufacturing lines for guide RNA. And since the guide RNA is required by the FDA and the EMA to be sterile, so we have grade C clean room equipped with grade A isolator to ensure that we can produce proper sterile products. And our facility and workshop has been designed to routine APS twice per year. So regarding our delivery track record, we have delivered till now more than 130 clinical batches, supported more than 40 projects, and 13 of them have got the global IND approval. Yeah, and the evolving regulatory landscape, this is our understanding about how the CRISPR evolves in the regulatory fields. So it is a very long journey for our clients, which is sponsors, to work from the very beginning of the discovery all the way to the commercialization. And the most challenging way for us as a CDMO vendor is the classification of this guide RNA material. At different application scenarios, for example, the ex vivo cell therapy, the guide RNA will be used with Cas enzymes and then will used to engineer the cells and finally injected to human bodies. While in vivo gene therapies, the guide RNA would be formulated with mRNA and then injected to human bodies. So we have different categories as the guide RNA as a starting material and also as a drug substance. So this is challenging for us to make it, to fit in all scenarios. And after the approval of the first therapy of Casgevy in 2023, FDA has a very clear guidance that the off-target would be the main considerations for the early discoveries. As for the components of guide RNA, we have very clear regulatory requirements, which is that the purity has to be greater than 80%, and any impurities that is greater than 1% should be identified. And also for the sequencing, this is a very crucial part since FDA is requiring that while using the sequencing method, especially for the next generation sequencing, to be used as a characterization method for the sequence identity test of the full-length products. And also it is required using this sequencing technology to be function as an orthogonal method for purity testing. And the last two items would be the residue solvents and also the elemental residues of the impurity testing, also about the stability test at the different storage conditions. And what we can offer to support the regulatory services. From the very beginning, our scientists will cooperate with the customers to design the sequences. We have some, guide RNA online design tools that can mitigate the off-target potentials, and also we'll have some, off-target analytical assays, including the IGAT, PAM6, so on and so forth, to predict or to evaluate the off-target risk. And through the IND submission, we can support any pre-IND FDA meeting, and also we will submit all the documents in a CTD format for the IND application. And during clinical phases, we can support also the amendments, including all the scaling up of the manufacturing process or the manufacturing changes, and also, of course, the vendor switching. If the customers are willing to switch from the current vendor to GenScript, we'll do the risk assessment and also all the supporting materials. And I'm going to share with two case studies. First is to use the NGS as a sequencing ID test. We know that the guide RNA synthesis is a very comprehensive and tedious synthesis routes comprising of over 400 chemical steps. And taking the capping, for example, if this capping process is not well tuned or well controlled, there might be a side reaction that will tune G to 2-amino A. And this grounding to 2-amino any substitution will not result in any molecular weight difference, because the molecular weight will be exactly the same. However, when we are looking into the base pairing, that this mutated base will no longer bind to C, while it will bind to T. So even if it's mutation of exactly the same molecular weight, the sequencing readout would be completely different. So it will generate a G to A mutation. And this G to A mutation will be very crucial because using conventional chromatography or mass spec analytical tools cannot tell the difference of in terms of chromatography retention time or the peak shape of the peak or the molecular weight are exactly the same. However, if we look into the right side of these slides, that when using the in silico prediction, we have if we have a G to A mutation, the potential of target risk significantly increased. And this is our internal data showing that how we tune this impurity control. So conventionally, if we overkill this capping reaction, that will observe more and more G to A mutation. So this is listed on the left side of the slides. And as we know that the mass spec and HPLC chromatography cannot distinguish the side purities, and this can only be read out using the NGS as a sequencing method. And if we tune the manufacturing processes, the G2A mutation can be significantly decreased. And this is the second case study of using the NGS as a purity analysis. We have a proprietary NGS library preparation workflow that we can utilize the UMI to characterize the guide RNA sequences. And since the FDA is requiring that the NGS is used as an orthogonal purity testing assay, we have shown 2 examples of the sequencing characterization here. Using NGS, we can report that the full-length product can be greater than 80%, and any impurities that is greater than 0.5% have been listed in this diagram. And what we can do using this diagram, that we can read out all the potential mutations, especially in the guide sequences. So we have read out all the potential mutations, and also we can distinguish an N-1 impurity. And the ratio has also been listed using the NGS testing. And using the in silico prediction, we can see that these impurities will have potential off-target risks. And to test that, we do some wet lab tests, including also a positive control, which is an off-target sequence. And we tested the editing efficiency of this one, of this set, and confirmed 2 of them as labeled in yellow to be as the crucial or critical impurities. And furthermore, we can do a risk assessment of using this, because when we spike in the synthesized impurities to the full-length product and do the cellular assay, and we can definitely see the significant off-target profiling of the 2 sequences. So this can help us to generate the risk assessment report and to submit to health authorities as a overall assessing, because the off-target is a major concern as of right now. And I believe that this is pretty much of my presentation, and happy to take any questions. Thank you. So if no questions, welcome to visit our booth at 5:15. So happy to look forward to further discussion. Thank you.